still waiting like 2-3 minutes, right? Before we start.
Yeah, we'll wait a moment for folks to roll in. Good morning. Welcome, everybody. Thank you for joining us for today's seminar. We're going to get started in just a moment. As you trickle in, a reminder that you can use our Q&A panel to ask questions. We'll hold questions until the end of the talk. You can ask questions at any time using that Q&A panel. So we'll give folks just one moment to trickle in for our seminar today, and then we'll go ahead and get started. Good morning, good morning. Welcome, everybody. Thank you for joining us. We'll get started in just a moment. As a reminder, please use the Q&A. Looks like 2 chat bubbles to ask questions at any point during the seminar. All right. So let's go ahead and get started. Take it away.
Okay. Hi, everyone. My name is Min-Hwan Kim, a senior scientist in the human cell type team at the Allen Institute. It's really a great honor and pleasure to introduce Dr. Brian Diaz today. Dr. Brian Diaz is currently an assistant professor in the Department of Pediatrics at the USC Keck School of Medicine and direct a research laboratory in the Developmental Neuroscience and Neurogenetics program at the Saban Research Institute, the Children's Hospital, Los Angeles. Welcome and enjoy your presentation.
Thank you. Good morning, everyone. I'd like to begin by thanking the organizing committee for extending me this invitation to talk to all of you today about some of the work that we've been doing over the last few years with the intent of really understanding and breaking legacies of stress. I should begin by saying that the talk does contain potentially sensitive imagery and dialogue, and so should that be upsetting in any way, please take care of your wellness as best as possible. In a postcard from the volcano, Wallace Stevens writes, "Children picking up our bones will never know that these were once as quick as foxes on the hill. A part of what it is, children, still weaving budded orioles, will speak our speech and never know, will say of the mansion that it seems as if he that lived there left behind a spirit storming in blank walls." The spirits of stressors and traumas not only storm the walls of the individuals who directly experience them, but also leave profound imprints in many ways, storming the blank walls of descendants as well. Legacies of stress and trauma can and do echo across generations. Take, for example, Rachel Yehuda's work, who's shown that babies who were in utero at the time that their mothers directly experienced the 9/11 terrorist attacks, and if those mothers went on to develop PTSD, at age 1, those babies had lower cortisol levels, lower stress hormone levels, compared to a comparable cohort of babies whose mothers experienced the 9/11 terrorist attacks but did not develop PTSD. And this is especially salient because Rachel's group has also shown that descendants of Holocaust survivors, even if those descendants did not experience the Holocaust themselves, had lower cortisol levels, and those cortisol levels were associated with a higher incidence of PTSD and anxiety. Then there's work by Tanya Jovanovic and colleagues who've shown that children born to mothers who were abused, and that abuse could have happened even before those children were conceived. When those children are brought into the laboratory, they present with higher anxiety and depression-like metrics compared to a comparable cohort of children whose mothers were not abused. And then finally, of course, we know through historical famines that have occurred, both anthropogenic as well as natural famines, that nutritional impoverishment in one generation has profound influences for future generations. What my lab is interested in understanding is, what do legacies of stress and trauma look like? How do they manifest, and how do we break them? I stand before you today as an ambassador for the work of everyone in the laboratory. This is the current team at Children's Hospital Los Angeles and the USC Keck School of Medicine, and we're really grateful to a lot of our funders as well. And I recently moved the lab from Emory University in Atlanta to Children's Hospital Los Angeles, and this was the lab at Emory. And again, I stand before you as an ambassador talking to you about many of these individuals' work today. So what my lab is interested in doing is, we primarily work in mice, and we're interested in understanding and breaking legacies of stress. While most of our work is done in mice, I'll end my talk today by talking a little bit about the collaborations we have in the monkey space, and then I've moved my lab explicitly to Children's Hospital Los Angeles to start working with in humans. So I have an ambitious agenda today, which is to share 3 stories with you. In the first story, which is really going to take place within a generation, we're not even going to address an intergenerational component. I'm going to talk to you about a brain region called the zona incerta, or the ZI, and its role in fear generalization using mice. In the second story that I'm going to talk to you about is, I'm going to talk to you about how we use olfaction, the sense of smell in mice, to really understand the phenomenology of intergenerational legacies of stress, try and dissect some of the causes, some of the consequences, and also the potential for reversing legacies of stress. And then in the final story that I'm going to be talking to you about, I'm going to leverage a collaboration that we have with Dr. Mara Sanchez at the Emory National Primate Research Center, which takes advantage of naturally occurring infant maltreatment in Rhesus macaques to identify mechanisms and biomarkers by which maltreatment really gets under the skin and becomes embedded in our biology. As I said, these are all the people who've done the work that I'm going to be talking to you about. The zona incerta work was done by Dr. Archana Venkataraman, who's now a postdoc at UCSF. The work on using olfaction to study legacies of stress was done by Hajawed and Soma Sunigrai and Sarah Hunter in the lab. And the collaboration with Dr. Mara Sanchez is really spearheaded by a graduate student in my lab, Laura Korobkova. What I'm not going to be talking to you about is our interest in the influence of glucocorticoid receptors on astrocytes and their role in learning and memory, something we've published on, and a new line of work in the lab, which is motivated by collaborations in the clinical space with our colleagues at Children's Hospital on Prader-Willi syndrome. So let's jump right in then into the story about the zona incerta and fear generalization. A big part of the lab is interested in understanding the neurobiology underlying dimensions of PTSD, post-traumatic stress disorder. And a debilitating dimension of PTSD is the lack of fear inhibition, the inability to put the brakes on fear. And one form that this lack of fear inhibition takes is fear generalization. And fear generalization is really the inability to inhibit fear to neutral stimuli that only resemble trauma-related cues. So for example, you might have a sound that comes to be associated with a traumatic event that has, that sound then comes to elicit a physiological as well as a behavioral response of fear. But fear generalization is the manifestation of that physiological response and behavioral response in response to a sound that had not been associated with a traumatic event. Why is this important? This is important because you could have a soldier in combat and they come to associate a loud noise with an especially traumatic event. And that is adaptive. But now that soldier comes back to their civilian setting, and every 4th of July hears fireworks, and they have generalized their fear to those sounds, resulting in the manifestation of a fearful response and of physiological responses associated with that fear. So what we'd like to understand is, how does fear generalization come about, and how do we put the brakes on fear generalization? How do we suppress fear generalization? So in thinking about how fear manifests, there's a lot of talk about learning associations, about stimuli and their outcomes. There's a lot of talk about recalling those associations and about extinguishing those associations. And so, of course, all of us have heard about things like, or brain regions like the prefrontal cortex, the amygdala, the hippocampus, playing really crucial roles in all of those memory-associated processes. But when we are thinking about all these processes, we also need to think about taking in sensory information and generating an appropriate motor output. And so when we think of it through that lens, we're thinking about stepping out of this canon, stepping out of the amygdala, hippocampus, prefrontal cortex trifecta, and looking at other brain regions that might be hubs for informational flow. And the thalamus and several thalamic brain regions now have been implicated in fear-related behavior. But in wanting to step out of even the thalamic circuitry, we were looking for a brain region that is involved in discriminating between specific stimuli and would allow for gating of specific stimuli to allow for fear to manifest or not. And so we trained our focus on the zona incerta, because the zona incerta, as has been shown in multiple publications several years ago, is involved in gating peripheral and sensory inputs and allows for discrimination to occur from a sensory perspective. And while we were ruminating about the zona incerta playing a role in fear generalization, there were two beautiful papers that came out talking about the role of the zona incerta in extinction learning and defensive behaviors, and the role of the zona incerta in memory acquisition. So Archana decided to ask a simple question: could the zona incerta be involved in fear generalization? And so the experimental framework that we use is a simple one to test fear generalization in mice. And so what we do is we bring animals to a particular context, context A. We present a CS plus that is paired with, in this case, a 0.3 milliamp foot shock. And we also present a CS minus that is not reinforced with any foot shock or negative outcome. In a completely independent cohort of animals, what we do is we bring those animals in and now train them, but pair the CS plus with a 0.8 milliamp foot shock, while the CS minus continues to not be reinforced with any negative outcome. And then what we can do is we can bring these animals into a completely different context and now present both the tones and test fear generalization. So what we found, and this has been really the bedrock, several others have noted this before us, is that training at high intensity foot shocks promotes fear generalization. So you have low intensity training and percent freezing on the y-axis. You see that the animals are freezing a lot to the CS plus, but negligibly to the CS minus, showing the ability to discriminate and not generalize their fear. But when you have high intensity training, you now see comparable measures of freezing in both the CS plus as well as the CS minus presentations. And when we measure something called a discrimination index, we see that low intensity training is accompanied by the ability to discriminate between the CS plus and the CS minus as a high discrimination index, versus the high intensity training has a low discrimination index and the animals are really freezing equally to both those tones. So the question then was, could the zona incerta be involved in fear generalization? And in the interest of time, I'm not going to go into it, but our hypothesis was activity in the zona incerta modulates fear generalization, and more specifically, increased activity of GABAergic cells in the zona incerta will decrease fear generalization. And Archana came to this hypothesis by doing a lot of activity-based mapping in animals using CFOS labeling to come to this hypothesis. And so to test this hypothesis, what Archana did was she looked at used VGAT Cree mice and dropped in dreads, Cree-dependent dreads, into the zona incerta. And so what we're doing here is we're dropping in the HM3DGQ G protein coupled receptor, which when activated, when the animals are administered CNO, those cells fire and are stimulated. So the question that we're asking is, does stimulation of GABAergic cells in the zona incerta prevent fear generalization? So the experimental protocol was take these VGAT Cree mice, inject the Cree-dependent dread that I talked to you about, or the Cree-dependent fluorescent protein control, train these animals at high foot shock intensities. So these animals would now generalize their fear, but now before testing, come and inject the animals with CNO, which would then activate or jumpstart the zona incerta. And so what we've put in is stimulatory G protein coupled receptors, the HM3DGQ dreads. And the hypothesis is that the zona incerta would not be active during fear generalization, which gives us fear generalization. But now when you stimulate those cells, you might prevent fear generalization. And so that's exactly what we see. So shown here, again, on the y-axis is percent freezing. Just like I showed you earlier, you take animals, you train them with high intensity foot shocks, and you see that those animals given CNO show a high freezing to both the CS plus and the CS minus. But when you have animals where you go in and stimulate the GABAergic cells of the zona incerta, you now see an increased freezing to the CS plus, but not as much freezing, negligible freezing to the CS minus. So now we've suppressed fear generalization in animals that typically show enhanced fear generalization by stimulating the zona incerta and GABAergic cells in the zona incerta. And that is also viewed as through the measure of the discrimination index. And then we followed up this paper by saying which projection patterns actually are involved in the fear generalization. So in supplemental figure 11 of that first paper that Archana published, we looked at tracing and pathways of the zona incerta to other brain regions that might be involved in fear generalization and extinction learning. And what we found is we chose to focus on, we found that it projects to the nucleus reunions, the thalamic nucleus reunions. We found that it projects to the periaqueductal gray, both brain regions that are crucially involved in fear-related behaviors. And we chose to focus on the nucleus reunions, projections of these GABAergic cells from the zona incerta to the nucleus reunions, because the nucleus reunions has been implicated in fear generalization from work from Tom Sudoff's lab, Steve Marin's lab, and many others. And so then what we wanted to ask in this paper that we published recently was to ask, do the GABAergic cells located in the zona incerta that specifically, and their terminals that terminate in the nucleus reunions, do those influence fear generalization? And so what we first did was we stimulated GABAergic cells in the zona incerta using optogenetics and recorded from reunions neurons, and found that stimulating these GABAergic cells induces inhibitory postsynaptic currents in the reunions, suggesting that there's an inhibitory pathway from the zona incerta to the reunions. And then what we did was we asked this question, do these zona incerta located reunion projections, the GABAergic cell projections, influence fear generalization? And what I'm showing you here is a similar experimental design to the DREAD experiment, but now here we're using optogenetics, where we're going in and putting in channel rhodopsin. We're stimulating these GABAergic cells only at the time that these animals hear the CS plus and the CS minus on testing day. And again, what we find is that shown to you in the pink dots is that, again, you take animals which have the Cree-dependent fluorescent proteins, so the optogenetic stimulation is not doing anything to those cells. And what we find is that there's fear generalization because there's increased freezing to both the CS plus and the CS minus. But now when you go and optogenetically stimulate these terminals in the reunions that originate from the GABAergic cells in the zona incerta, we find that there's the freezing to the CS minus is still high, but we see a plummeting of freezing responses to the CS minus, suggesting that there is now a suppression of fear generalization. In the interest of time, what I'm not going to be talking to you about is the same paper. We show that there's an effect on extinction learning, which is another form of learning that's associated, deficits in which are associated with PTSD. So what I've done in this first story is I've told you about how GABAergic cells in the zona incerta in adult mice, so this would be considered within a generation, when we can induce stressors using high-intensity foot shocks, how GABAergic cells in the zona incerta actually are able to modulate fear generalization, and how we can suppress fear generalization by stimulating these GABAergic cells, and specifically their terminals in the nucleus reunions. I'm now going to go into the second story, and this is where I'm talking about intergenerational legacies of stress. And remember, I'm talking to you about this through the lens of trauma and stress existing in various forms, like I talked about. The 9/11 terrorist attacks, the Holocaust, famines that have occurred across history, domestic abuse, and the fact that these traumas echo across generations. The question is, how does trauma and stress echo across generations? And to be clear, we were not the first ones to be studying this phenomenon. A lot of individuals, beautiful work by Dr. Tracy Bale, Dr. Isabel Mansui, Dr. Ollie Rando, and many others have taken work in rodents, and they've manipulated either diet or they've manipulated psychosocial stress parameters in an ancestral generation of mice, and then looked at future generations and seen how that initial stressor, be it nutritional, be it psychosocial, how does that permeate, how does that influence future generations that may not have been directly exposed to those insults. But the message that I take away from all of those studies also is that all of us are looking for a needle in a haystack. Where do you look for the imprints of psychosocial stress, of nutritional insults? And so my job, or my thought was, we need to shrink that haystack and make the needle that we're looking for much bigger. And it occurred to me that we could use the power of olfaction, the power of smell, to be able to tag a stressor in one generation and then follow it across generations. And the reason for that is, of course, we know that the mammalian olfactory system, there's an exquisite structural and functional specificity to it. So within you and I, in our noses, reside olfactory sensory neurons on the main olfactory epithelium. And those olfactory sensory neurons send axons into the brain that terminate in the olfactory bulb. Now, an olfactory sensory neuron, or an OSN, can express 1 out of 1,000 different odorant receptors that are encoded in the genome. So in panel B from this paper by Di Maria and I, what we find is that of all the colors of the rainbow that olfactory sensory neuron could express odorant receptors for, it only expresses the red receptor. Of all the colors of the rainbow that second neuron could express, it only expresses orange. And what's exquisite about the olfactory system is no matter where in the olfactory epithelium these olfactory neurons reside, all the olfactory sensory neurons that express the red receptor send their axons into the brain, and they coalesce into one unit at the level of the glomerulus in the olfactory bulb. And why this is important is because we have mice in the laboratory, and I'm showing you M71 lag-Z mice, where we can stain the M71 expressing odorant receptor olfactory sensory neurons as blue. So where the MOE is, you can see dots, which are puncta, which are the olfactory sensory neurons that are expressing M71, and they're sending their axons into the brain, and those axons are coalescing at the level of the olfactory bulb. We also have MOR23 GFP mice in the laboratory, and those mice, the MOR23 neurons are activated by LIRA. The M71 lag-Z mice, the M71 neurons are activated by acetophenone, an odor that smells like almond to me, but has been referred to as a cherry blossom odor. And propanol, another odor, does not activate these M71 neurons. So then it occurred to me, as I said, that we could use olfaction to study how legacies of stress could be bequeathed to future generations. And the way we do that is we tag stress in one generation using olfaction. So we take a particular odor, say acetophenone, Acetophenone plus mild foot shock, acetophenone plus mild foot shock. We do that 5 odor shock pairings, and we do that three days in a row. So 15 odor shock pairings in total. And then because the olfactory system has functional and structural specificity, maybe the olfactory neuroanatomy that I gave you a primer on and the behavior to that particular odor may be affected in the future generation and may be a legacy of that olfactory stress. And maybe the odorant receptor genes may bear imprints of that ancestral experience with the odor. And so we've done this work now over 3 papers over the last several years, and I'm going to share with you salient points from each of those papers. So the experimental design is take the F0 generation, the parental generation, and expose them to olfactory fear conditioning. So odor plus foot shock, odor plus foot shock, odor plus foot shock. And then the comparable control animals get exposed to the odor without any foot shock. And then we allow for mating to occur. We remove the males before the pups are born, so they don't have any interaction with the pups, and then we test the descendant generations. So what I'm showing you here is our data from the initial paper that we published in 2014 and have since replicated in 2 more papers since. What we find on the y-axis, you see % odor potentiated startle to acetophenone. We say that that is a sensitivity to acetophenone. And what we find is that F1 animals, and I'm referring to the middle bar right now, F1 animals in black that are sired by males that have been conditioned with acetophenone, those animals, even though they've never had exposure to acetophenone before, are most sensitive to acetophenone. Whereas animals that have been sired by F0s that were conditioned to propanol, those animals do not show a sensitivity to acetophenone. Now, when you look at the graph for the % odor potentiated startle to propanol, you see that animals that have been sired by F0s that were conditioned to propanol, those animals show a higher sensitivity to propanol, whereas F1 animals sired by F0s that were conditioned to acetophenone do not show that sensitivity. I want to reiterate that point, that there's a behavioral sensitivity to the conditioned odor in the F1 generation to the odor that the F0 generation was conditioned to. I do not refer to that as a fear of that particular odor, but a sensitivity to that. And I stress that so much because this will become important when I talk about what the consequence of that sensitivity is in the 3rd study that I'm going to be talking to you about. So we have this behavioral sensitivity to the F0 conditioned odor in the F1 offspring that have never experienced this odor, been exposed to this odor before. What does the neuroanatomy look like? So here we're looking at M71 LAC-Z animals, and this is what the glomerulus looks like in F1 animals that are sired by F0 mice, male mice that are hanging out in their home cage. When you look at F1 animals that are sired by F0s that have been conditioned to propanol, remember propanol does not activate the M71 receptor. There is no change in the olfactory glomerular neuroanatomy. But in odor-naive animals, F1 animals that have been sired by F0s that have been conditioned to acetophenone, there's a dramatic expansion of neuroanatomy that is devoted to the M71 receptor. And since this paper, in the subsequent papers in biological psychiatry and in genes, brains, and behavior, we've shown that this is not something specific to M71 LAC-Z and acetophenone, that pairing, but we know that we've shown that it also extends to, for example, MOR23 GFP and Lyral, which is an odor-OR combination. So what I've shown you thus far then is that tagging stress in one generation using smell allows us to follow the behavioral and neuroanatomical imprints of that stress in offspring, such that when you take animals and stress them with a particular odor, they become, the offspring, even though they've never smelled this odor before, become sensitive to the odor and there's an enhancement in their olfactory neuroanatomy. The question is, how is this happening? And so to really delve into mechanism, there were two main possibilities. One is there's a transmission of information, no different than social narrative, social transmission of information that occurs. So the male might have conveyed some information to the female, and that results in the sensitivity and enhancement in neuroanatomy during mating. Or there could be an inheritance of information via the sperm. In the interest of time, I'm not going to go into these data, but what we did is we did in vitro fertilization. We looked at the F2 generation, and we did cross-fostering studies. And so in all of these cases, the effects, the behavioral sensitivity, as well as the enhancement in olfactory neuroanatomy persisted. So now when you have in vitro fertilization, where there's no normative mating that's occurring, there could be no social transfer of information that's happening during the mating process, and only sperm are fertilizing the egg, and we still see those effects. That to us implied that there was an inheritance of information from one generation to the next via the sperm. Now the question becomes, do sperm even register information about the salient olfactory experience? And so we harvested sperm from these animals, and then Active Motif did DNA methylation analysis around these odorant receptor promoters. And the reason for that is BLUE is blue for an M71 lag-Z neuron in an F0 generation, and BLUE is blue for an M71 lag-Z neuron in an F1 generation. What we thought was happening was we were making a lot of blue or a little blue, a lot of blue or a little blue. And so we invoked epigenetics into this discussion. And at the risk of simplifying things, we know that all of us have DNA, which is a book of life. And that book needs to be read in many different ways. Depending on how that book is read, you're going to get very different meanings. So take, for example, this rather ominous sign. And then you take this sign and put a much needed and strategically placed full stop, and the meaning of that sign changes. That's essentially what we have within all of us. We have readers, writers, and erasers that put the equivalent of these punctuation marks like DNA methylation, like histone modifications, like RNA modifications, that then allow for genes to be expressed or not. And these readers, writers, and erasers serve to link the environment with our genome via epigenetics to allow for environmental experiences to now interface with our biology and allow for gene expression to be orchestrated. And so what we did was, as I said, we looked at the sperm of these F0 male mice, and Active Motif did the DNA methylation analyses around these odorant receptors in those sperm. And what we found is that in sperm harvested from F0 ace males, these are males that were conditioned to acetophenone, there was less methylation around the OLFR 151 receptor compared to the methylation around the sperm, around the OLFR 151 gene in the sperm of the F0 propanol animals that have been conditioned to propanol. And this was specific to the OLFR 151 receptor, because when we looked at methylation around the OLFR 6 receptor, which is not activated by acetophenone, we find that there's no differential methylation across these two groups in the sperm. So what these data suggested to us is they added to the story by saying that we can tag a stressor in one generation using smell and then follow that stress across generations and offspring. And what we find is that not only is there a sensitivity to that odor or smell in the offspring, not only is there an enhancement in neuroanatomy for that particular odorant receptor population in the offspring, but the sperm of that ancestral generation register information about that particular olfactory experience. So the questions we then wanted to ask were cause, what causes this? How does this come about? What does this do for the next generation? What consequence does it have? And can we reverse it? And to address this cause and consequence question, what we did was we published a paper in Genes, Brains, and Behavior that addresses both of these components. When we think about DNA methylation, as has been widely reported, it may not really be the mechanism by which information can be inherited from one generation to the next. And that is because of the phenomenon of epigenetic reprogramming, where methylation marks are scrubbed clean just after fertilization and when the primordial germ cells are developing. Although there's now data to suggest there are some loci that do escape epigenetic reprogramming, we're trying to understand what is the logic behind those loci escaping reprogramming. But what we postulate or posited in that Nature Neuroscience paper is that RNA found in sperm might act as conductors of gene expression. And when we sent that paper out for review, one of the reviewers said that is not how biology works. But we posited that RNA in sperm might act as conductors of gene expression because RNA in sperm could then influence the zygote, which is depicted here on Waddington's landscape as this ball at the top of a hill. And as that zygote develops, it could hit rocks on the left-hand side and land in one place as the adult develops from that embryo, or the ball could roll down the right-hand side and hit a bunch of rocks, but also go down several ravines and end up in another place as an adult that develops from that embryo. And so what we do now is we inject RNA from sperm of F0 order A plus shock males into naive single-cell zygotes, and then put those zygotes back into surrogate females, get those offspring, and then look at their anatomy and look at their behavior. And what we find is that we can recapitulate all of our effects that we had seen. So we can recapitulate the sensitivity to odor. We can recapitulate the enhancement in olfactory neuroanatomy when we inject this RNA from F0 acetophenone plus shock or F0 lyral plus shock males into single-cell zygotes. Unless you think this is something that is science fiction, we were actually one of the last to come to this experimental table. Beautiful work again by Tracy Bale, Isabel Mansui, Chi Chen, as well as Ollie Rando, have now shown that you can take RNA from sperm of male mice. They might be subjected to psychosocial stress, they might be subjected to nutritional insults, and then do the same experiment that we did, and then they recapitulate all of their effects just from that RNA found in the sperm from those stressed males. And so RNA in sperm seem to be messengers of intergenerational influences of stress, and now there's a burgeoning literature to support that. So that's one way by which the baton of stress is handed over from one generation to the next, at least on the paternal lineage. Then the question becomes, what effect, what does this pre-existing sensitivity do for the next generation? And when we think about intergenerational legacies of stress, there's always talk about the gloom and doom that comes with it and the detrimental influences of intergenerational legacies of stress. But what we decided to ask is, are there any positive consequences that come from this? And the way we decided to ask this is we did the same experiment as we did before and got the F1 generations. But now what we do to the F1 generation is we expose them to single odor plus shock olfactory fear conditioning. So just one odor trial pairing. And then we look to see the next day, do the animals remember this association of odor plus shock? And what I'm showing you here are graphs on the top as well as the bottom, which show that because of this pre-existing sensitivity to the paternally conditioned odor, the future generation is able to remember and consolidate information about the single odor trial, odor foot shock conditioning compared to the animals that did not have this pre-existing sensitivity. So it's as if the pre-existing sensitivity is priming the olfactory system to now pay attention and consolidate information about sub-threshold events that are associated with this particular odor. So from this part, what I've shown you then is legacies of stress perpetuate in part through the paternal lineage by RNA that are found in sperm, and that there are some positive consequences that we have not yet appreciated about legacies of stress. And one of those positive consequences might be that the system, the biological system, is being primed to respond now to contexts and cues in one's environment that it would not have been able to respond to if it did not have that previous legacy of stress. In the final part of this story, I want to talk to you about reversal. And this was really motivated by a question that was posed to me by retired General Pete Chiarelli, who said, what would you like me to do with this information? Not enlist anyone in the US Army whose parents or grandparents suffered from their own PTSD anxiety. And so the question that we wanted to ask is, has the ship sailed, or can we reverse and halt the influences of stress by intervening in the exposed population? There's a wealth of literature on being able to go into the F1 generation with environmental enrichment and several other interventions and reverse or halt legacies of stress from perpetuating. The question is, has the ship sailed, or can you go into an F0 generation that has experienced a stressor, intervene in some way, and halt those legacies from now going to the as yet unconceived generation? So what we did in this paper was we took the same experimental approach. We take animals that we train with the odor, so they're acetophenol plus foot shock, or Lyral plus foot shock. The control animals are just exposed to those odors. And then we have something called an extinguished group. So the extinguished group is where we stress the animal with an odor. We make that animal fearful of that odor, but then subsequently expose that animal to extinction training. Extinction training is now exposure to that odor with no foot shock. And so we're calling this the extinguished group. And so in pictorial form, what we found is that once again, we find that when we have, compared to the exposed animals, the F1s of the trained animals have an enhancement in olfactory neuroanatomy. There's an enhanced behavioral sensitivity to that odor that the F0 was conditioned with. And there's that imprint in the F0 sperm at the level of DNA methylation. But when we take these animals who had been previously made fearful to the odor, so they were trained, but now we extinguish that response, we find that we no longer have the behavioral sensitivity to that odor. We no longer have that enhancement in olfactory neuroanatomy, and we no longer have that DNA methylation signature in the F0 sperm. So what I've shown you then is that the die is not cast. What this suggests is that there is a malleability in the system such that when you have a book that is edited in one way, we can re-edit that book in different ways. And in that lies our ability to halt legacies of stress and trauma from perpetuating across generations. So from the olfactory use of olfaction to study this phenomenon, what I can tell you is that future generations bear sensitivities to the odor, in this case, that F0 was conditioned with. Their brains are organized differently by enhanced neuroanatomy for that particular odorant receptor. And the sperm bear of the ancestral generation register information about a particular experience. Sperm RNA, or RNA in sperm, are carriers of information that allow for the baton of stress to be handed from one generation to the next. But it's not all gloom and doom, because there are some positive consequences of intergenerational legacies of stress. And one of those in our case is that you're better able to consolidate information about specific cues in your environment. So what I want to do now in the last 5 minutes that I have with you is I want to now turn my attention to a naturally occurring early life stress that occurs, infant maltreatment in the Rhesus macaque, to really test this idea about what are mechanisms and what might be biomarkers that allow for an insult like infant maltreatment to really get under the skin. And so the research question that we're asking is, are there signatures of early life stress that we can detect in biological specimens that could then point us in the direction of mechanisms of intergenerational legacies of stress, but also hold the promise as biomarkers? And so we wanted to hold our feet to the fire here in some ways, and not only use blood as a potential biological sample, because the criteria for the specimen that we wanted was it might want to be, we wanted to be a readout of biological perturbations at the scale of the entire organism. And so enter exosomes and extracellular vesicles, which are blebbing off from all tissues in us, and they're entering into our circulation. And so we think that these exosomes then, which are carrying DNA and RNA and proteins, could be harbingers, a good beacon as a readout of what's happening organismal-wide in the shadow of something like infant maltreatment. So we get exosomes, we perform RNA-seq and then look to those profiles to say, are there biological signatures of early life stress? And so to do this work, as I said, this is a collaboration with Dr. Mara Sanchez at the Emory National Primate Research Center, spearheaded by Elise Morin, Hassa Wallam, and Laura Korobkova. And what we're doing is we're leveraging this naturally occurring infant maltreatment that you're going to see in the next slide, which is happening in semi-naturalistic environments in outdoor runs at the Emory National Primate Research Center. So this naturally occurring maltreatment occurs in 2 to 5% of the population. There's physical abuse that occurs. There's a high amount of infant rejection. It's very intense during a critical period of infant development, which is the first 3 to 6 months of life, and it's repeated with successive infants. That is, if maltreated females often maltreat their own infants. And so here's what that dragging looks like. Whoa. And here's what rejection looks like.
He sure is. God.
And so that happens, as I said, naturally. And so we got plasma from infants that have been maltreated as infants or that are raised with care as usual from their mothers. We get RNA from those exosomes. We extract RNA from those exosomes which are in the plasma, and then we did RNA-seq. And I'm just going to show you some Go analysis that we performed where we think that these data suggest, so first thing what you'll see is that there's far more gene classes, far more biological processes that are suppressed in the maltreated infant exosomes compared to those that are activated. And when you look at those, it seems to us like perturbations to immune function and mitochondrial energetics are one potential mechanism and could be used as a biomarker for understanding how infant maltreatment actually gets under the skin and might have deleterious consequences for those infants. So what I've done thus far then is I've shared with you 3 stories today. In the first, within a generation, just adult mice, making a case for the zona incerta and its projections to the thalamic reunions in fear generalization. The second story, leveraging the power of olfaction, of smell in the mouse to really tag a stressor in one generation and follow the intergenerational legacies of that stress that occurs via RNA in sperm, that has adaptive consequences for the offspring, but that can also be reversed. And then shown you studies that are unpublished thus far, profiling RNA in exosomes from monkeys who've been maltreated as infants versus those receiving standard care, and some biological processes that might be allowing for that maltreatment to get under the skin. Once again, these are all the people who've done this work. I am deeply grateful to them, deeply grateful to my current lab and all the funding sources. And I find that there's way too much shade thrown on people's work these days. I don't believe this and I don't believe that. These are only some of the people who inform and inspire my work. I stand on their shoulders. I thank them, and I thank you for your attention.
Oh, thank you very much. It's a great talk. I think I didn't see the question from the Q&A, but if any audience have any question, and feel free to ask directly or just write down in the Q&A. Yeah, I saw one question in the Q&A in Martin Lohmann. And can you comment more on later generations?
Absolutely. Thanks for that question. So we've only looked till the F2 generation in the olfactory legacy of stress, and it goes to those grand offspring as well. So that's the generation that we've looked to. We don't know if it propagates even beyond that. The second question is, can oocytes transfer the information as well as sperm? And I think this leads to that, is related to the third question, which is, does the stress trace propagate from mother to offspring? The answer is yes. In that first Nature Neuroscience paper, we showed that both the paternal and the maternal lineage can pass this information on to future offspring. We have not yet targeted the oocytes and tried to get at mechanism and what's happening in the oocytes to allow for that information to go through to the next generation, but it definitely goes down the maternal lineage. And we know how important maternal zygotic RNA is. And so undoubtedly, there's going to be a big influence of oocyte RNA, I would imagine, in the propagation of intergenerational legacies of stress. But that's a gap in the field in general, which we're all trying to now fill because of the technologies that are coming to the fore with being able to work with lesser and lesser number of eggs and do those kinds of analyses. Would use of gabapentin or similar medications assist in fear neutralization? So I think that's an interesting question. I'm not so sure about the pharmacology of it, because we know that drugs that affect GABAergic signaling do influence fear behavior, but they also come with their own other side effects like sedation, etc. And so we don't want to numb the system as well. And so I think there's a trade-off there as to what drugs one can use. How large do you think the space of intergenerational messages can be? For example, in this talk, you highlighted 2 orders that could be communicated specifically and separately. So I apologize for not understanding that question as well as I should. When you say space, if you mean a sensory space, there was a reason why I chose olfaction, because I could hypothesize that there might be an epigenetic signature in how odorant receptors might be regulated that might result in this legacy of olfactory experience being passed on from one generation to the next. And so if I would think about it in a sensory space, I couldn't think about that happening in the auditory space, for example, but we know that it occurs in the gustatory space. And now we know, of course, from work in worms, for example, that pathogenic avoidance can go multiple generations as well. We've seen the olfactory system and the intergenerational legacy of olfactory conditioning in fruit flies as well. So I think it's sensory modality specific, and not all sensory modalities will allow for that baton of stress to be passed from one generation to the next. But feel free to maybe clarify if space wasn't the sensory space as intended in that question or answer. Does the observation of olfactory stimuli sensitization in F1 generalize to visual stimuli or other stimuli? So thank you for that question. I didn't answer the visual stimuli aspect. I think it's a really important one. I can see where visual stimuli could also go, because again, you're selecting for rods versus cones with certain sensitivities, etc., on a spectral profile. One thing I should mention in that regard is that some people have taken this work and talked about it through the lens of, is this how instinct comes about? Is this how the first fear of a particular odor that we now call hardwired in a mouse or a rat, is this how it came to be instantiated in the biological system? Other people have talked about mate preference in guppies, for example, which is color-based, and how did that initial mate preference come about? That's out of my wheelhouse. I'm not an evolutionary biologist, but I just wanted to place that in the context of people are talking about this work in the context of instinct. Are there any intergenerational transfers of positive experience? I think that's such an important question, and our data are limited on that in so much as you think, for example, cocaine is a positive experience and self-administering cocaine is a positive experience. There are data, beautiful data from Christopher Pierce's group and Fer Vasile in particular, showing that the self-administration of cocaine does have intergenerational effects from a sensitivity perspective. There's less work from a parenting perspective. But I think those like work like Regina Sullivan's, for example, are really tapping on the door of that particular question. And I think it's early days, but one that we're watching very closely and are sure to provide answers. Odor paired with a reward instead of a shock. Absolutely. I don't know the answer to that question. I know that there are some studies that are happening in that arena. I'm consulting about those studies. It seems to be promising, but not something that I can share just yet. Is there a general understanding on what kind of information could be propagated and what not? I think that's such a beautiful question. And the way I take it is, what is the quanta and qualia of information? Can all information be propagated across generations? If that's the case, we'd have a signal to noise problem, I feel. And so there has to be some threshold. I unfortunately can't answer what that threshold is, other than to say, I think it needs to be extremely salient for the organism. And at least in the mouse, I can speak for the fact that taking these mice that live in standard housing and then giving them an odor paired with a foot shock is extremely salient, which is why I think we're getting some information from one generation to the next. But I think it all depends on what our definition of salience is and the threshold beyond which that particular environmental event or cue bypasses that threshold or not. Do sperm or oocytes happen to be particularly efficient to sensing circulating exosomes? So this is work really pioneered by Ollie Rando and Tracy Bale and a few others, where they've now shown that as the sperm exit before ejaculation, they go into the epididymis. And in the epididymis, there are these epididymosomes, exosomes hanging out in the epididymis that are chock full of small non-coding RNAs. And it seems like the RNA is shoved into sperm at the epididymis through these epididymosomes, and then before ejaculation, now those sperm carry these RNA, and that's what's giving the information to the next generation. I apologize, I don't know as much about the egg as we know about the sperm from those studies.
Thank you very much.
Thank you everyone for all those questions. It looks like we have gotten to the end of the questions. So thank you again. This is our last distinguished seminar of the year. And we will see you again for our next seminar in January.
Thank you, everyone.
Thank you.
Thank you.